Thanks very much. It's a real pleasure to be here today in both such an amazing venue, but also with such an interesting lineup of speakers. I think we're going to have a really, really interesting day. So as Claire said, my name is Anne Phelan. I'm the Chief Scientific Officer at Benevolent. And over the next 20 minutes or so, I'm going to be talking to you about our current thinking in terms of target identification, but also take something of a look into the future around where I think artificial intelligence augmentation can really have a benefit and an impact in the target discovery space. So for those of you who don't know Benevolent, we are a clinical stage AI-enabled drug discovery company. We have a portfolio of assets, everything from the very earliest exploratory phase of a drug discovery portfolio all the way through to phase 2 assets. And clearly we've got AI-augmented capabilities at all elements of the drug discovery process. But for the purposes of today, I'm going to really focus in on target ID. We at Benevolent have spent a lot of time investing and thinking about the very best way that we can come up with means of identifying really the very best target. And in lots of ways, it is the single biggest decision that we make in drug discovery. If fundamentally you've chosen the wrong target at the outset, then everything else down the line gets increasingly difficult in terms of getting to the clinic and getting any chance of clinical success. So underlying everything we do at Benevolent are our data foundations. We take a view that if you really want to start to understand the complexity of human disease biology, we need to be able to find, ingest, organize, and make available as much biomedical data as we can possibly access. And when I talk about biomedical data, there's some categories here on the slide. We're thinking about literature, so published literature, patents, any form of written documentation, where large language models can increasingly extract not just facts, but also inferred knowledge from that literature. We've got information around the pathology of diseases, symptoms, imaging information, but also really a focus on biological systems. So we're thinking about dysregulated processes and mechanisms of disease right at the cellular level, thinking about intracellular interactions, signaling cascades, mechanisms and processes, any one of which may be dysregulated in a very cell-specific manner to manifest as disease. We've also brought through a range of different experimental data types, so assay data, CRISPR, whole genome CRISPR knockout sort of information, iPS-derived data. We have a lot of omics information, so not just looking at the genes, the proteins, the isoforms, but also looking at transcriptomics, genomics, and that kind of real patient-first, patient-centric assessment of data to really get to the heart of those dysregulated mechanisms causing disease. And of course, we've also a whole massive range of entities and molecules, antibodies, drugs, other elements of pharmacology. And really, in building this knowledge foundation, we made a very conscious decision right at the outset to bring in this great diversity of multimodal data, because we're acutely aware of the limitations and biases of every single data type. So the logic is that by bringing together all of these different modes of multimodal types of data and looking at the interconnectivity of that information, we're able to really maximize our understanding of human biology as an entirety and really start to break down the silos across therapeutic areas to connect shared mechanisms across diseases. One of the things I've discovered whilst working at Benevolent is really, in the past I've worked in particular therapeutic areas and I've looked very specifically for targets in that space. Whereas actually, when you take away those silos and you think about human biology and human dysregulated processes, there's an amazing commonality across diseases for dysregulated mechanisms in different cell types, different organs, and the way they manifest in disease. Ultimately, what we've built by ingesting all these different multimodal data types is a proprietary integrated view of biomedical data that we believe supports our ongoing decision-making. Now, clearly, there are challenges with bringing such a broad range of data modalities together. And we know that every single type of data we consider has its limitations. It's incomplete, it's noisy, it's contradictory, it has particular hierarchies, particular structured and unstructured characteristics. So to overcome this kind of hurdle, we have built a range of data processing pipelines each one of which is specifically designed to be able to ingest and unify particular types of data so that we can integrate them in a unified data model system into our integrated knowledge graph. And an example there, so we've got structured data, obviously, that's comparatively straightforward when you're looking at ontologies with hierarchy. Unstructured data is more complex, so I'm talking about literature, patents, clinical trial data. And the way we process those is using our natural language processing pipelines, where we're able to take named entity recognition to highlight and identify key elements of any written text to build it into sort of a structured, multidimensional architecture of data and information. Similarly with genetic genomics data, so we're looking at GWAS, single-cell RNA-seq data, epigenetic data, clinical data. We have a bespoke data processing pipeline for that too, where we're looking, for example, we have a Spark-enabled GWAS pipeline. We've got whole exome and whole genome pipelines, where we're able to ingest these large data types, layer over them our variant to gene annotations, and then again build that back into our data infrastructure so that we really have a great diversity of data coming into this holistic picture. Insights from these sorts of primary data, also when they're integrated alongside the published findings, really create this orthogonal view of human biology. With this, we're sort of constantly building this mechanistic landscape of disease. We're thinking about the dysregulated processes and mechanisms in our quest to find really what we think is the very most relevant target for potential therapeutic intervention. So this slide just gives you an overview of our target identification process. So considering that we've got our data architecture in place, we've generated our pipelines that allow us to process, unify, and integrate the data, we then run through this high-level process. So we have built this knowledge graph, and the drug discovery scientists at Benevolent have the capacity to interrogate and interact with the data to really start to explore disease. We're looking at things like specific cell types, dysregulated mechanisms, particular tissues, and we can start to formulate a picture of disease biology, irrespective of the disease, to understand the best way really to tackle that disease from a drug discovery perspective. We then have a suite of algorithms that I'll come on to shortly that enable us to predict targets based on this foundational data knowledge graph. We then take these targets, predicted targets, through a rigorous target assessment process where we, as drug discovery scientists, are provided with a lot of supportive evidence to help us understand how the AI-augmented models, what data they have used in order to predict these particular targets and surface them to us for assessment. Once we've gone through the assessment phase and we've got the list of targets down to a very high quality, shorter list of targets, we take them through to experimental test and validation. So this whole target identification process culminates in what we regard as a validated hypothesis. So it's a data package where we have high confidence that this target, with a high quality drug, will have the potential for therapeutic value. To give you a bit more colour as to the way the drug discovery scientists would interact with the data, we sort of think about it as posing a biological question to our suite of AI models. There's a very simplified version of a question there. Can we treat chronic inflammation in ulcerative colitis by reversing immune cell activation in colonic mucosa? So in crafting this sentence, we're pulling through a disease, a sign, we want to reverse the effect, we have a mechanism and a tissue. And this really gives the models key focal points in which to pivot in order to generate the potential target hypotheses. These relevant data from the whole wider knowledge graph are linked to these biomedical concepts in the question so that we get a really integrated picture of the disease. And the nodes that come to the top really are high quality predictions. I want to emphasize that this is a really rapid, iterative, repeatable workflow, and it allows us to iteratively explore the type of question, the type of mechanism, the type of cell for any given disease, and really refine that biological question. Once we got the questions, we were able to capture all of our decision-making in a very consistent format, and this allows the intent of the scientist to be recorded within the platform, but it also creates a sort of virtuous cycle of learning and decision-making within our Benevolent platform. Once we've gone through this process, we have got a well-crafted, we believe, question with which to pose to our AI-augmented tools. And again, because we are looking at lots of different data types, we have lots of different elements in the question, this requires us to have multiple machine learning models as part of our predictions. So you can see there, each of the different AI models runs over a different particular type of data. So we have large language models. Obviously, they can extract information from written text, but they can also increasingly infer new knowledge based on published information. We have graph completion models, so these are very much the inferred relationships where we know certain facts and we can infer the next stage of a pathway or process. And we have particular models running over genetic, genomic, transcriptomics models. And again, we're thinking here very much at the patient level, looking at individual single-cell RNA-seq, looking at, as Klaus referred to in the introduction, looking at very particular regulated and dysregulated processes, looking at predisposition to disease, considering the disease, the genetic landscape in which a disease manifests. So we've got these specialized models, and they are designed to produce complementary predictions based on each of the individual data types. But really, what we want at the end is the very shortest list of high-quality targets that we can take through our process. So we've spent quite some time developing smart aggregation models. These allow us to really, based on our prior knowledge, our decision-making, what we understand about the disease exploration that we've undertaken, it allows us to bring to the surface the very best quality targets that we think have the highest probability of success. I mean, it's fair to say at this stage, the targets that are predicted are a mixture of comparatively well-known all the way through to really entirely novel, unknown targets with respect to disease. So our capacity to really think about the best way to get the mix of targets and aggregate has been a really focused effort for us this last couple of years. Ultimately, once we've gone through this process, it results in a list of potential targets for expert triage. And the Benevolent platform provides, from a drug discovery perspective, a really nice interface for us to undertake systematic review of these potential targets. So we have a tool with which we are surfaced with an awful lot of supporting evidence. It speaks to the underlying logic as to why a particular target was predicted and prioritized by our tools. And we take a very systematic view of the targets. So we're looking at the biological rationale. Why do we think this target, if modulated, would have a therapeutic value? Would it be safe to modulate? To what extent is this novel or is it well known? The more novel things are, the more important it is to be able to build the circumstantial evidence around the biological rationale. Very practically, we have to think about progressibility. If these targets come into our portfolio, we have to know that we can take them all the way through to the clinic. We always have an eye on precision medicine opportunities. Is there a patient-centric view of this target or this disease that we need to be considering? And then druggability. Really, what is the best modality? It could be a small molecule, it could be an antibody, it could be any technology. So keeping an open mind at this stage as to the best way to prosecute these targets. So once we've got these, we've made these decisions as to whether we want to progress a target. The decisions are all captured, again, in a structured and unstructured manner. And this allows us to have both an audit trail, but allows the system itself to learn over time. Once we've gone through this process and we've been able to triage the targets and get those ones we think are really the most relevant, they progress through to experimental validation. Now, this experimental work could be done in our labs in Cambridge, in the UK, or we also have a network of CROs and academic collaborators. And really what we're doing at this stage is using whatever modality we think is the most relevant in order to best test the hypothesis. It could be small molecule, it could be siRNA or CRISPR. We might undertake these studies in iPS-derived cell lines, in primary patient-derived tissue samples. But really what we're trying to do is give the very best opportunity to test the hypothesis and make a decision as to whether or not we think this is a viable potential path forward. The diversity of experimental data that we generate similarly further enhances our representation of human disease, and we're in this constant learning loop. Once we've gone through this process, we've generated the target hypotheses, we've triaged them and experimentally validated them. We have to make a decision as to whether or not that target ought to come into our drug discovery portfolio. So this just gives you a snapshot of our portfolio today. You can see we've got everything from targets in early discovery all the way through to phase 2. And as we feel very strongly that this portfolio of assets really underwrites the approach that we're taking to take this multimodal approach. We're looking at a disease agnostic format. We're looking at modality agnostic assessment of our targets. And it's repeatable, and we continually build our portfolio based on this path forward. But so we feel like we've made really good progress. We've built this massive data foundation. We have very sophisticated models to reason over all of those data to come forward with these predictions. But actually, we still have a long way to go. This is a very relevant quote from Sidney Brenner way back in 2002. We're drowning in a sea of data and starving for knowledge. I think, as we look forward now from into the future, the next 5, maybe 10 years, we know that we have the capacity to generate more and more data on an exponential scale. But really, our capacity to reason over those data and really extract the best knowledge and value is still a work in progress. So this may seem very practical. I don't see on the horizon a fantastic eureka moment where everything just gets solved overnight. I think progress in drug discovery foundationally doesn't change. And this is exemplified by AstraZeneca's 5R framework that was published back in 2014. And it speaks to the right rights. We need the right target. We need a strong link between the target and disease. We need to understand predictive biomarkers. We need to make sure that we're targeting the right tissue. We get the drug to the right exposure, the right site of action. We need to be sure that these targets, when modulated, are going to be safe. We need clear margins. We need to understand selectivity and off-target effects. Obviously, we need to get these drugs into the right patients, make sure that we have a good risk-benefit assessment, and we also have to be aware of the right commercial opportunity. We're increasingly looking at personalized healthcare strategies. So all of these elements need to come together in order to get the right drug. And I think what is on the immediate horizon for us, because of our increased access and the increased availability of biomedical data, together with ever-increasing improvements in machine learning algorithms, and perhaps crucially, increases in our computational power. Now more than ever, we have the capacity to really ask drastically more complex questions and queries of the data at our disposal with real-time iteration. And I think the consequences of these 3 convergent elements is that, of course, we're going to get an improved understanding of human disease and human biology, and that will inevitably lead to optimization in target identification. But it's going to impact drug discovery across the board. Every element of drug discovery is going to move forward in this constant incremental progress. Clinical trial design, patient diagnosis, early signs of efficacy, they will all benefit from this sort of revolution on the immediate horizon. So in terms of technology investments, where I see things moving in the next 10 years, as I said, I don't think it's going to be a single eureka moment, but I think every element of these 5 rights is going to, in itself, move on substantially. Thinking about the right target, large language models now, sort of OpenAI, are fantastically improving our capacity to reason over huge amounts of data to really pull through the most salient and most relevant facts. Binding pocket elucidation, so the AlphaFold work recently published, again, gives us the opportunity to consider every target. We've got epitope definition, alternative modalities. In every way we can think about the right target, we have advances absolutely on the near horizon that allow us to prosecute those targets more effectively. In terms of right tissue, now single-cell RNA-seq, really understanding the expression of genes, the regulation of genes at a single-cell level as part of a wider tissue is something that is coming on unbelievably quickly. Access to iPS cells so that we get patient-derived cultured systems representative of tissue biology, high-content imaging, understanding targeted delivery. All of those are helping us to drive towards this right targeting of tissues. Similarly with safety, genetics plays a big part in safety, looking at traits, health records, CRISPR, omics data. all of it together really gives us a picture of the potential risk-benefit profile of targeting any particular gene with a potential therapeutic. And similarly, the right patients. So again, genetics and omics, as Klaus highlighted, this is really in the process of a revolution in terms of volume, processability, but also the extent to which we can extract information. So again, the expectation that we will have a much more patient-centric view of drug discovery in the now and near horizon is an absolute. And again, the commercial opportunity, we're looking increasingly at optimized clinical trial design, precision medicine, diagnostics. So I think the totality of all of this data that's coming through now, all of which is augmented through AI and machine learning tools to enable us to really extract maximum benefit and information from the data available to us, is really on the brink of a revolution. So looking ahead, I think our capacity to conjure with these right characteristics simultaneously is going to be the key. We can't be thinking about find the right target, then wonder about the next tissue, and then will it be safe? What patient will I dose? This has to be done simultaneously through multidimensional analyses. And it's inconceivably complicated for a human, but actually this is exactly where AI and machine learning models really come to the fore. And I think they're going to bring a step change in our ability to treat diseases. As I said right at the outset, choosing the right target remains the single most important decision that we make. But I think increasingly there's going to be the context of that target for each patient and the therapeutic opportunity of that target to treat or cure that patient and the way that we consider therapies leading towards a genuinely personalized approach, again, is something that I think there is an expectation that the drug discovery world will deliver this for patients. And in addition to this more comprehensive understanding of disease, the vast array of targeting modalities that are becoming mainstream now really broaden out what's feasible for us. Now, it's fine to identify the best target, and you can really sincerely believe this is the best target for that disease. But if you don't have a modality with which to target it, those very valuable targets sit on the sideline. So I think now this increase in broadening out of what was previously perceived to be the druggable genome will enable scientists to really target the very best genes identified, irrespective of the modality required. So I think I'm out of time exactly. I just thank you for your attention.